Hey everyone. The title might have made it seem like this episode's going to be a downer, but thankfully, we have our old friend Jason Dombroskie from Cornell leading this one. And his enthusiasm for our six-legged friends is as infectious as Steve's giggles. You might recall Jason from our Insectapalooza episodes. Well, he reached out this past summer and kindly offered to join us for another episode. We batted a few topics back and forth and landed on the insect apocalypse, something Steve and I hadn't heard of, but it's just what it sounds like, the crash of insect populations worldwide. There's a lot of debate among entomologists about the details. What exactly is going on? How severe is it? And most importantly, what can we do about it? So in part one, we head out on the trail with Jason. He'll introduce us to some insects we find along the way, and he'll school us on why insects are so important. And in part two, he delves into the details of the insect apocalypse, what we know and what we don't know. And since we feel bad that you can't see what we're getting to see, we bring back a little trick we had in our last episodes with Jason. Each time we find a critter, listen for the sound of a camera shutter. That's the signal to visit this episode's page on our website, thefieldguidespodcast.com, and we'll have photos there time-stamped so you can see what we're looking at, along with some extra info. Enjoy! Hello, and welcome to the Field Guides. I'm Bill, and I'm here with Steve. Good afternoon, Steve.
Good afternoon, Bill.
And we're also here with Jason. Good afternoon, Jason.
Hi, gents.
And what we're going to do today is give you the experience of what it's like to be in the woods, in the field, and on the trail. Each episode, we pick a natural history topic, research the science behind that topic, and then take you out to a natural spot and share with you everything that we've learned. And this is one of Steve and my favorite kind of episodes because we have had to do virtually no research for this one because Jason here, who has been on the podcast before, Jason Dombroskie from the Cornell Insect Lab, he was kind enough to reach out and invite us to come on out here to the Rattlesnake Hill Wildlife Management Area. And we are going to be talking today about what, Jason?
We're going to talk about the insect apocalypse. Ooh. A real buzzkill.
Jason's wanted to ruin our day. You know.
Is that pun intended?
Ooh, it wasn't. But yes, I'll take credit for that one.
Well done, sir. Well done. So, folks, Rattlesnake Hill Wildlife Management Area. Steve is going to give us some quick background on where we are today.
Off the top of my head, this is a state-owned 5,100-acre tract right at the edge of what most would consider western New York. It's managed by the DEC. It has some hardwood forests, some wetlands and grasslands, and provides habitat for deer, wild turkey, ruffed grouse, and waterfowl. I wasn't reading anything.
Thank you, ChatGPT.
Yeah. Main uses here are hiking, bird watching, horseback riding, cross-country skiing, and hunting and trapping.
And I actually have a story about this. We were talking before we turned on the mic, how difficult it is to find information on this. This is, for us, from...
What doesn't the DEC want us to know? That's what I'm wondering.
For more populated areas of New York State, this seems to be kind of out in the middle of nowhere, right?
Yeah, very much so.
And it's, as Steve just said, it's kind of right on the border of what most people would consider western and central New York. So between Buffalo and the Finger Lakes, not too far from Letchworth State Park, if people know where that is. But I have never been here before. I don't think any of us have been here. No. So we wanted to pick a spot that was kind of in between where Jason lives and where we are. So we landed on this, and it rang a bell when you mentioned it, Jason, because 25 years ago when I was working at Beaver Meadow, actually my first day there, I think, I was being introduced to some of the visitors by Dave Junkin, who we've had on the podcast. Oh, yeah. He was my boss, and he introduced me to one guy. I don't want to say his name, but he did not seem very impressed with me right off the bat. And because I wasn't a birder, and he was a hardcore birder, and he could tell I knew virtually nothing about birds. And I could tell he was thinking in his head, why the hell is this guy? And Rattlesnake Hill.
Hey, hold on. Isn't the logo or the animal for Beaver Meadow a beaver?
It used to be.
There's no way it's a bird.
It is a bird now. Because they're owned by the Buffalo Audubon Society. God. Yeah, they changed it to a wood duck.
They should change the name of the place. The Wood Duck Nature Center. Wood Duck Meadow. Sorry.
So anyway, so the name of this place came up, and I said, oh, where's that? And I still remember the disdain in his voice. You've never been to Rattlesnake Hill? I was just like, geez, what's with this guy?
If he's still with us, you should message him and let him know you finally made it out. Yeah.
He is still with us.
We're now officially part of the club, I guess.
Yeah, I guess so.
He's no longer angry. Or maybe he will be. I don't know.
It just took me aback, like, whoa, dude. Like, Take it easy. So I'm interested to see what we find here.
Well, I think before we start, you've got some lovely equipment you're holding there. You're not just going to hold it and look pretty.
Oh, yeah.
Why don't you describe what you've got here? I'm going to actually put you to work.
I got an insect net. I think I've only caught a leaf so far.
Well, that's a good start. So that's just an aerial net, so typical butterfly net you're picturing. That's for capturing insects on the wing or insects on the ground. Try to avoid any raspberry brushes or multiflora rose. I see some there, so try to avoid that to not rip my net. But one thing I always do when I bring people out with a net is I make sure they've actually used one before, because I'm surprised at how people think they know how to use the net, but they don't actually.
I'll tell you.
So can I insult your intelligence here?
Yes, please.
Do you know how to use a net?
I've had to do it for a job once because we would do these insect sweeps along our transects for our plant surveys. But I wonder if there was some detail that I forget, because I was just swooping left, right, left, right, and I would do it all along maybe like 20 or I don't know, 10 meters or 50 meters or something.
Yeah, and that was done with a different net called a sweep net.
Okay.
That had much tougher material that can take vegetation.
It was definitely not this net. This is you could tell a flexible.
So like this is more for dragonflies and butterflies and things like that in the air.
Okay.
So
the answer is no.
Well,
I don't know.
You had most of it there. And the trick is, when you get something, when you actually catch it, you get it into your net, and I give it an extra few sweeps to bring it to the bottom of the net, and then with a flick of the wrist, roll it over, and that captures it there. The next thing to realize is once you've got something, the thing is going to want to fly up. So don't go, where is it?
Right.
And look down at it and have it fly up into your face.
Yes.
You want to pinch it off and sort of look at it like that, and sort of laterally, so it's not going to get away on you.
So Jason's grabbing like the last 6 inches of the net and kind of making a little bag at the bottom of the net. Yeah.
Exactly, yes. Yeah. And you can look at it there, or you can transfer it to a vial to get a clearer look and whatnot.
All right. So just a few things I want to clear up before we move on. One, Steve mentioned that this property is operated by the DEC. So we've said it before on other episodes, folks, but the DEC is our New York State Department of Environmental Conservation. And then I feel like we should just, again, give people a quick intro to you, Jason. You've been on the podcast before.
Yes.
But just tell us very quickly, what's your job? What do you do?
So I manage the Insect Diagnostic Lab and the Cornell University Insect Collection. So basically, I identify stuff for a living that people submit to us. They pay $25. We do our best to ID it. We also do IDs for various state governments and used to be federal government contracts. So I won't get into that. Do our best to ID those. And then the collection part of it is I manage about 7 million dead insects and make sure they stay in good shape. And we're continually adding to that collection.
Yeah, so we did a 2-part episode a few years ago where Jason had us out. We got an incredible tour of the insect lab, and then in part 2, you took us out mothing in the evening. So if you haven't listened to those ones, folks, go check those out. Yeah, I can't recommend them enough. So Jason's knowledge is encyclopedic, and you are a fantastic science communicator.
Well, thank you.
So, yeah.
I make it up as I go.
All right, so we are out here in the field today. Jason was saying that there's kind of an open area as we enter into this trail, but folks, we are out here on a day. It's about 70 degrees. It is overcast. It rained yesterday, and it may rain on us today, so I don't know how many insects we're going to see.
We'll at least talk about what we can't see.
That's right.
Because they're here still.
And that will fit into our topic of the insect apocalypse, right?
Exactly.
Yeah, it can also be the theater of the mind. They can't see anything, right? They're like, we're covered in insects right now. We just start lying to you guys and you wouldn't know.
That is true.
Yeah.
True.
That could be a delusion, though, too. That could be a sign of other issues.
The episode, we lost our minds in the field.
So where we're standing right now, we're on kind of a pretty overgrown trail here.
Looks like good tick habitat,
It does look like very good tick habitat. And we're standing in what looks like a relatively young mixed forest. So probably less than 50 years old, wouldn't you guys say?
Yeah, you got some early successional species here. Lots of black cherry. There's a white birch over there.
Yeah, white birch tells you it's pretty early.
Some young red maples. Looks like mostly native species, though, which is nice.
Yeah. All right, let's see what we find. Now, we mentioned that this episode is going to be about the insect apocalypse. So, Jason, do you want to kind of give people a quick and dirty rundown of what that means?
Yeah, so basically it's the short answer is most entomologists have a strong feeling backed by some evidence that insects are declining dramatically. And in some places in the world, very dramatically, which I'll get into a bit later. And this can have some very serious effects because insects are really quite important. In fact, if I only put you guys on the spot, why would you consider insects as important?
Well, a huge one is pollination, of course.
Pollination, yeah.
So without pollination, we're not going to have food, right?
Yep, exactly.
Now, it's all obviously not run by insects. They're done by insects, but they're a huge part of it.
And I believe it's 60% of all vascular plants are pollinated by insects. So,
Wow.
And that's not just crop plants. That's, everything around us, minus a lot of these lower plants here. Now, actually, let's stop right here because we do have some insects flying.
We do.
Is it tiny moth I'm getting out of the way?
Well, we can just look at it. It's
Okay.
sitting nicely for us. This is a type of moth called a snout moth. Oh, and another one just sat right next to it. So this one, you can see they're long and skinny. They've got a big, long snout, which is actually part of their mouth parts, the labial palps.
Now, hang on. You're saying big and long. This whole moth is what? Less than an inch, from stern to tip, right?
Yeah, you got to understand. My sense of scale is skewed.
Right.
I work on very small moths.
Oh, didn't you say in the last episode you really like, or do you work with micro moths?
Yes.
Yeah.
And that is a micromoth.
Okay.
Yeah. And so this one is obviously adapted to grassy habitats. It's long and skinny. It's streaky looking, has that snout which extends its profile. And indeed, this particular species does feed on grasses. It lives down low near the base of the plant. And the caterpillars are actually very rarely seen because of that, because they're really down near the soil.
Okay.
Some of the related species can be pests and turf situations, like sod webworm, but in nature, you just see good numbers of them. And this is one of the more common species here.
And what was that one called again?
The vagabond crambus.
Oh,
I love it.
Agrophila vulgivagella.
wow. What does crambus refer to?
So crambus is sort of that old genus that all of them were lumped into.
Okay.
I guess more proper name, you could call it a vagabond grass veneer. That's sort of the more British term for the crambine moths that this belongs to, or the snout moths. But yeah, so there's actually quite a diversity of these in New York State. The crambines, that subfamily, some are really habitat specific. So there's some that you only get in bogs amongst sphagnum. Some you only get up sort of higher elevations like in the Catskills and Adirondacks, where they're associated with sort of dry rocky mosses and things like that. So, but yeah, this is one of the common weedy species you can get in any city, any place you pull off the highway, any place where there's low vegetation. This moth will be there.
Is there a range like pretty much across North America? Or?
I think it's more of an eastern thing. I don't know how far west it goes, but
Gotcha.
yeah, it's all over New York State.
All right. So, like I said very briefly, it was small. What would you say? Less than an inch across, right?
Yeah, absolutely.
Yeah, and just kind of cream colored, right?
Yeah.
So how did you ID it so quickly on the wing?
So it comes with experience. I know that if I flush a small moth that size in this habitat in August, it's going to be probably Agrophila, one of the two common species. The other one, Ruicalis, is a bit smaller, little slightly different markings. And then if it's early in the year, there's a bunch of other species it could be. So it's experience.
Yeah. You have the search image, but you also have the context.
Yes.
To put it in place.
Yeah, and it just comes with, a lot of it is experience. If I was in Nevada right now and I had the same thing fly by, I probably wouldn't be able to identify it.
Okay.
I would have to catch and go, okay, it is that.
Yeah.
Right.
You'd look for the big long snout.
Yes. But on pollination, generally, if you had asked me a few years ago, is this a pollinator? I would have said, it might visit flowers, but probably not. However, there's been recent studies that are showing that things like raspberry in the UK, while bees visit the flowers more, moths actually carry a heavier pollen load when they visit them at night. And this moth, I've actually seen visiting goldenrod at night, usually the early part of the night. And so it could be an important pollinator. We just don't know.
Okay.
Wow.
All right. So that brings up two things in my head. One, you're talking about moths and coming out at night. And I'm sure some of our listeners, if they haven't listened to the previous episode, they're in the questions popping up in their head, differences between butterflies and moths. So I suggest people go back and listen to our original 2 episodes because we cover that in depth. It's not as simple as you think. And then the second thing was, in the limited research I did do for this episode, the one thing that came up referring to the insect apocalypse is, you just mentioned that we're learning new things about this moth, is that there are definitely research gaps, especially
in terms of the tropics and the southern hemisphere, where insect diversity is abundant, but we don't know as much about the insects that live there to say, yes, there's definitely an insect apocalypse happening there. Is that, would you say that's accurate?
Yes. So our knowledge gap is huge even here. Okay. So, and I'll give you an example of, generally for bigger, showier stuff like butterflies, we tend to know them pretty well. However, just a few months ago, a new type of swallowtail was described from upstate New York.
What?
Something that's been under our noses for all these years. And a bunch of us had an inkling, like, that's different. There's something slightly different about it. And it took now, it's tough to identify, let me tell you. The big difference is the flight period and slight difference in host and whatnot.
The way this one looks, because I thought maybe I came across this, is it one that has more like black wings with a white stripe on it, or am I thinking of it?
No, like it's much more subtle than that.
Okay.
It's a whole table of usually has, usually bigger, usually smaller, but it's called the midsummer swallowtail, Papilio solstitius, after the solstice. It was actually described by an amateur entomologist from the Rochester area.
Whoa.
And he paired up with a bunch of people more heavy in the genetics end of it, and they published this new species recently.
Wow.
Cool.
And so even the big showy stuff, right here, where we've had entomologists working for hundreds of years, 150 years. Wow, really stretched it there. Much less species than the tropics. We still don't know what's going on here. Now, when you get to the small stuff like the micromoths, it's just daunting how many, new species there are. But yeah, the tropics, it's absolutely embarrassing what we know.
Right.
And what they do in the environment. Like, a few years ago I said, no, that's not a pollinator. And I was like, well, I've seen it visit flowers at least. So.
I have a question that I told Bill I was going to ask you, and this is a good time to ask it. Now, I've always heard little stories here and there about, like, we have this species, but the only place that we've ever seen it is in this one collection. We only have a single individual, and we've never seen this ever outside of this collection. Mm-hmm. Have you come across that personally, or is this something that you've just heard of other collections having, or other entomologists?
I remember reading a number of years back that something like, I want to say 20 or 30% of insects are known from a single specimen.
Wow.
I don't know
like
how
1 out of 5?
Yeah.
Possibly.
Yeah, especially with the tropical stuff. So at Cornell, we've got a lot of material from southeastern Brazil, from the Atlantic coastal rainforest from the early 1920s. Mm. And a lot of them are singletons, and that habitat's essentially gone. Like only 10%'s left, and that's an area of high endemicity. So a lot of species found just there that no doubt are gone, and we might have the only records of, but we just need more entomologists to work on it. Yes, I've definitely come across singletons, both things I've collected that I can't match up with anything.
I've never heard of singleton. I like that.
I'm trying to think of the context that I use that in. I guess, when you're sorting traps, you have singletons, and those are always the gems.
Good question, Steve.
Yeah.
All right, so let's keep moving.
Yeah, and so you mentioned pollination, which obviously they're important in both agricultural systems and also in natural systems. Insects are very important. What else? Why should you care about insects?
Steve?
Why should we care about insects? Outside of pollination, surely they're a huge food source for a lot of other animals as well.
Absolutely. Biomass, that's another huge one. So right now we're getting into a more mature forest here. This looks like mostly sugar maple here. So a fairly young sugar maple forest, but this type of forest, about a month or two ago, would have been full of caterpillars. There's still caterpillars here, but the peak definitely corresponds with when birds are breeding here. And those caterpillars make up very significant biomass here. In fact, I used to love, there was an illustration on one of the trail guides in Algonquin Park in Ontario, Bat Lake Trail, that has an illustration of relative biomass of animals.
Oh, I've seen this one.
Yeah, isn't that a great figure?
Yeah.
So what it is, so first of all, if you took all the animals in a typical forest like this, typical hardwood forest, put them in a big pile, and looked at the relative mass, most of that, about 80%, it's going to be caterpillars and other insects, but mostly caterpillars. Then everything else is like a rounding error, like amphibians, there's a few. Birds, some of those. Mammals,
they're like the smallest. One of the smallest.
And then once you get to the top predators, like bears and wolves, don't even mention it. They're not significant.
I think I referenced that picture in our redback salamander episode, because I think in that the redback salamanders did take up a surprising percentage of the biomass.
Yes, in that particular system, yeah.
Yeah, and they like that. Their biomass was bigger than white-tailed deer or something like that.
Oh, yeah, and it should be way bigger than that, just by trophic level. And so for trophic level, basically it's when something eats something or gets energy from something, and every time you jump a trophic level, you lose energy.
Yeah,
and
a lot of energy.
Yeah,
it's like 90%.
So if you look at total biomass for the forest around us now, most of it's plant material.
Yeah.
And then there's going to be quite a bit of fungi as well, especially in the soil. And then insects will be less. Things that eat those insects, which are also insects, less. And then you get, the vertebrates, the insignificant things.
Right.
We're not that important, right?
Yeah,
in the big picture. Yeah. John Eastman, who we talk about all the time, his book of Forest and Thicket, it's always stuck with me in his introduction to the book of Forest and Thicket. Mm-hmm. Matt's mentioned this on his podcast, In Defense of Plants, about John Eastman talks about the plants are the power base for everything. And they often get short shrift in terms of celebrity status, but in terms of biomass and importance, like the plants and those producers.
Yeah, and it
huge.
definitely changes through the year too. So in the winter, there's a lot less insect biomass. There's still actually quite a bit in the soil, but above ground, it's pretty sparse. Yeah. And then it beefs up again in the summer. And that's why we've got these birds that are flying all the way from Central and South America up here for just a few months to reproduce because of the abundant food.
It's worth the risk of that trip.
And just to throw some numbers on it here, I'm terrible with numbers, so I got to look at my cheat sheet here. So in a study done in the Brazilian Amazon near Manaus, they had 1 billion invertebrates per hectare.
Oh my God.
That's a big number and, small, but that's 200 kilograms dry weight animal biomass in a hectare. And 93% of that was invertebrates.
Wow.
So 93% of the biomass.
So insects are huge.
Yes.
Yeah.
And then of that, now in the tropics it's quite different than here, where, a third of that invertebrate biomass is ants and termites. Now, there's probably no termites here. There's one species in New York, in upstate New York, which it could be here, but they're pretty rare. Ants, there's probably quite a few ants here, especially in the soil, but not like in the tropics.
Now, come on, I've had many people tell me they have termites in their house up here.
That's nice.
That's what I say.
Prove it.
Right.
We get sent a lot of quote-unquote termites into the lab and a lot of quote-unquote bed bugs. And, well, with bed bugs, it's about a 30% successful identification rate. So.
No.
Well, folks, I will try to find that picture because I have a lot of back issues of the Raven, and I bet I could find that picture and post it in our episode notes.
Yeah, and if you can't find it, I think the Friends of Algonquin Park have the trail guides available online. And you can see the cover image there, so you might be able to find a link to it there. If not.
I don't think we said, you talked about birds and the birds that were here in the maples during the breeding season. Folks, we are here in mid to late August, so it's August, what today?
21st.
21st. So you're not hearing much birdsong around us right now. There's a little bit, but they're pretty much done breeding. Maybe only goldfinches are still on eggs right now or raising young, but the birds are pretty much done breeding, so they get to be quiet.
And also, as I mentioned, coincides with a quiet time for caterpillars.
Yeah.
So there's a lot less caterpillar biomass right now than a few months ago.
Right. Yeah, a bunch of our warblers have already left for migration. So, all right, let's keep going. We're heading uphill right now. All right, so we know that...
I just want to... Something that came up earlier today. I heard that just when you think of weevils alone, like, there's many, many, many more species of weevils than there are, like, of all fish species.
Yeah.
Yeah, like, by a lot. Like, by tens of thousands.
Well, and even caddisflies. Like, who thinks of caddisflies? They're a relatively minor insect order. There's as many caddisflies as there are birds.
Which is amazing.
Wow.
Yeah. It's just, but everyone's just looking at the birds and the fish, and but in terms of species diversity, anyway, they they pale in comparison.
So insects need more publicity, right?
Yes.
They're not as for most people, they're not as charismatic, right?
They can be cute though.
Oh, I agree, but I think we have a skewed perspective compared to most people, right?
I loved when I saw spider pads blowing up on the internet. Because a lot of people were comparing them to kitten feet. Do you know, like the pads on the bottom of dogs and cats' feet?
Yes.
Spider pads kind of blew up, at least in certain areas of the internet,
Yeah, and it's just like, some of the furry moths blow up on occasion. And,
oh, yeah.
Butterflies really get most of the publicity, right?
Well, those are moths in that case. So.
All right, so we're going through some tall stuff here.
And definitely getting a good soaker.
A lot of goldenrod, some dogbane.
Oh, and here we have some bees. Ooh. So speaking of pollination, there's some bumblebees hunkered down on some Joe Pye weed, which is a common plant here, a native plant. And it looks like deer herbivory on parts of this Joe Pye weed. Okay. But the bumblebees are just chilling because it is kind of cool. Beep. Oh, you denied it. It lifted its leg.
Last episode we had a guy named Sandy on, and he's the one who taught us during one of his classes that you can pet bees. And it's actually kind of cool.
Steve is petting the bee right now.
He tried to get me. I've never had one try to get me before, but he's just chilling, you know. He just didn't want to be bothered for that second.
Yeah, so there's something that obviously is an important pollinator for certain crops and a lot of native plants as well. But, you know, there's a lot of research that can be done with insects. So if you think of anything in biology, what doesn't involve Drosophila at some point? You know, that is one of our standard lab animals. A lot of studies in evolution involve insects because they're easy to grow. There's lots of them for now, you know. So because you have such diversity, you have so many possible systems to study. So, also very important in scientific research. And then along the lines of pollination and agricultural services, so a lot of them are beneficial because they're eating pest species. One of the best defenses against insects eating your garden is to have other insects eat those insects, and that's a whole thing in itself. But they might also be declining as well. And I think I'd be remiss to not mention that, you know, insects can have a negative effect on humanity. So invasive insects we'll talk about a fair bit today. Where am I here?
The spotted lanternfly was just on the news. My parents asked me about it, and they're like, "Do you know about this thing?" And I'm like, "Yeah."
I stopped on the way here, and I saw a spotted lanternfly monitoring trap, and it was empty, luckily. But,
they're cool looking, though. You know.
So
kind of like how a starling is cool looking.
Yeah, no, they're very pretty, and they'd be especially pretty in Southeast Asia where they're native to. But here they're a bit problematic. So another big one is disease vectors. And I'll throw another phrase at you here. Throughout history, more soldiers have died due to diseases spread by lice and mosquitoes than from bullets and bombs.
Wow.
So think about that. You know, like we've got all these fancy weapons, but the insects are much more efficient at killing us. And fleas, you know, can spread things like the bubonic plague, and that killed 50 million people, you know, so insects can have a real negative impact on humans as well. But
they're real terrorists.
Oh, Steve.
But it's the minority of species that are problematic.
Right.
You know, when I like to tell master gardeners where I do training sessions for them that if you took the insect biomass in a good, healthy garden, a small percentage of that will be what you'd call non-beneficial insects. So problematic insects that spread viruses or eat whatever. A small percentage will be beneficial insects where they eat those negative insects or they're pollinators or decomposers or whatever. But most of that biomass is going to be just neutral insects that are just, they're there.
It's like a classroom of second graders. No, I'm just kidding.
Wow.
Just kidding.
You know, I think you're an insect of a person, actually, for saying that. You know? They're our future, Bill. All right? Just
the key thing is, though, you know, we have to realize that not everything has to be good or bad,
right?
You know, and that's hard to get across to people at times. Where it's like, oh, is this a good bug or a bad bug? Does this insect bite? It's like, well, anything can bite.
Right.
You know, like, if you pick me up, I'll bite you.
Hey, even Jason can.
Some insects don't have mouth parts.
Fair enough.
They can suck.
They don't even have those.
That's true.
Some of them only live for what's the type? There's one that only lives for many that probably only live for a few days in their adult forms.
There's some that live a few hours. Some of the flies as an adult.
Shout out to our very first episode, the peacock fly, right? That does the goldenrod galls. Do they have mouth parts?
I believe so. They probably do.
Never mind.
They're probably just imbibing fluids, though, as a supplement. Because with most insects, most of the feeding is done in the larval stage or the nymphal stage. But of course, there's exceptions to everything.
But to get back what you were saying about good and bad insects, it depends on context, right? It's not clear cut.
Yeah.
Also, I'm sure some insects are bad for maybe for goldenrods, for example. But I like them. So I think they're good.
Well, in a place like Europe where Canada goldenrod is a nasty invasive,
nice.
Anything that eats goldenrod is fantastic there, you know?
Sure.
Yeah.
Whereas here we have this huge fauna of insects adapted to goldenrod. In fact, if you look around the goldenrod, you can see bunches of leaves tucked together.
Oh, yeah? They got the cool flower galls.
Yeah, and that's a type of fly called a cecidomyid, a gall fly. And going back to diversity, there was a DNA study where we looked at just DNA barcodes and just sort of lumping them together by DNA similarity. And it estimated that there's probably more types of gall flies than elder flies combined.
Wow.
That we probably know less than 1% of the species. Like, it's just daunting.
Yeah.
And how many people are studying gall flies in North America?
Probably not many.
Raymond Gagne, who retired a bunch of years ago. And I think that's it.
Oh my gosh.
Seriously?
Yeah.
So there's almost nobody.
Essentially, unless there's someone that I don't know about. But
Because isn't even this one, is this one also a gall? I thought I had read that this is like a coin-shaped gall.
So Steve, tell us what you got.
So I just have a goldenrod leaf right now, and there's a little black circular spot on the leaf that has a kind of like a white ring around it. And I think for the longest time when I was younger, I was like, oh, this is just like a fungus or whatever. But later on, I think during the gall episode, we had talked about this, and we said maybe in a future episode we'll talk about some of these other types of galls.
So you're actually half right.
Oh, is there a fungus involved with the...
Yes, there's a symbiosis between a gall maker and a fungus.
That's cool.
So I believe it's called Asteromyia. It's another cystomyid midge. And there is a specific fungus to that particular type of gall.
Wow.
And so, for each insect, there's a smaller insect or a fungus,
Right,
so
right.
it's just, the diversity is never ending.
But when we see the same thing on or we see a similar looking thing on something like a horse chestnut, that one actually is like a disease, not just a gall. It's not even a gall at all, right?
In North America, there's some fungi that will cause blotches on horse chestnut. In Europe, there's actually leaf miners that can become very, very common.
All right.
Maybe someday we should do it.
We should.
Visit the goldenrod galls and talk about a bunch of the ones we skipped the first time.
It's been 10 years.
Yeah, right. We just redo all of our episodes, but with the stuff we didn't cover.
More bumblebees here. Actually, quite a few of them on some of the goldenrods. Just hunkering down, waiting for a bit of sun to warm up. So one of the challenges with demonstrating insect decline is getting quality data and trying to sort of establish a way to census how common insects can be. And that's been one of our biggest challenges in entomology. Because when you think about it, we were in the past sort of going after the rare stuff, going and collecting all the really rare things, ignoring the common stuff, and not using standardized methods to survey things for the most part. And now we don't have that data to work with as a baseline. And so, I think of the human brain is bad at... Sorry, I got distracted by a moth there. That's one of the bird poop mimic moths there. This is Olothrudis bipartitana, which looks like a piece of bird poop. It is quite small, so that's, about a centimeter, so half an inch. And sort of banded gray and white. And if you look at it not expecting a moth, it does look like a little piece of bird poop.
It sure does. I would walk right by that. It's just on a leaf,
cool.
Edge of a leaf.
And actually, I can use that one to illustrate why insect sensing can be challenging in the first place, and that is identification. So my expertise is in that group of moths, leaf roller moths. And I remember back in grad school when I had a bunch of moths that looked kind of like this. And I read through the literature, and I was like, okay, I've got these sorted. I know what these two species look like, and okay, I've got this. And then someone said, are you sure? You should really confirm. Okay, so I luckily had an undergrad that was volunteering with me, and she dissected about 50 specimens. And my success rate was about 33%.
Wow.
I had 5 species in 3 genera. And it just did not match with how they looked.
Wow.
And so basically for identifying these moths, you have to dissect the genitalia because they're often really complicated structures, and that's what you use as sort of the gold standard. You can also use DNA methods to identify them, but yeah, for this particular group of moths, the genitalia are really quite distinct.
Wow.
Now I have a good feel for some of them with experiences like, okay, I know that if it's completely pale in the outer half of the wing, it's almost certainly bipartitana. It's going to be that one. Whereas all the other ones have some sort of marking there. And then once you get into the other ones, then it's kind of gets into guesswork without a dissection. So yeah, it can be challenging to work with old data because you don't know if species have been described since then, if they were good at identifying things,
Wow, yeah.
So it's,
Right.
It's not like birds where you can be like, okay, well, they had a sparrowhawk, which, oh yeah, that's this, and
Yeah.
oh, we've actually split that species, so we kind of have to throw away that data, but with insects, it's just way more daunting. And the other thing, I read an interesting paper recently describing something called shifting baseline syndrome.
Yes.
Yeah, did you read about that?
Well, I teach about that in my wilderness class.
Oh, okay, yeah. And it reminds me of many discussions I've had with my dad over the years.
Oh, right. Because everyone's born into their own new normal, right?
Yeah, and that's the normal. And I still remember, my dad looking up at a gull flying over and saying, "Oh, when I was a kid, there was never any gulls up here." It's like, yeah, because when you were a kid, DDT had wiped them out.
right.
But your dad probably knew gulls, and so it's, we have that sort of normal that we assume. And the other problem with our sort of the meat between our ears is that we're bad at remembering the normals. We remember the extremes. So, you always have the stories of, I remember when I drive and, the whole windshield be coated in insects. It's like, well, maybe so, but maybe that was one or two or three times.
Right.
That's what's stuck
It's
in
like, was that every time? Or,
not every time. And maybe, it's not all biomass is created equally. Maybe you're driving through an agricultural field and there's a big pest emergence,
Sure, right.
which is very different than, let's say, some common native species that's emerging huge numbers or mayflies or something like that.
I wonder if anyone's ever tried, and maybe this would be a horrible idea because I haven't really thought it through just right now, is if anyone tried to do a survey right before cars went into a car wash, like set up at a Delta Sonic somewhere and just, get permission to do your survey there, and just detect how loaded windshield bumpers or windshields were or something.
With insects.
Insects.
Yeah.
What would you?
I didn't know what you were saying.
Anything loaded with anything.
Yeah.
Sorry.
So, folks, to circle back for just one sec, when Jason mentioned shifting baseline syndrome, it's an interesting concept to look up because it applies to more than just natural systems, but it's just the idea that each successive generation accepts current conditions as normal. So with each successive generation, we get away from previous baselines. Mm-hmm. So in terms of biodiversity, it's changed a lot since people came from Europe here to North America.
Yeah.
Like the baseline has shifted drastically. I've heard some people refer to the impoverization of the ecology of North America. We look around and things look beautiful, but the Everglades, which we try to visit every year, bird populations there, 90% of them, that's what the decline has been.
Wow.
Since the 1800s. Now you see 10% of the birds that used to be there, and the bird life there seems incredible, but compared to what it was, 100 or 200 years ago, it's 10%.
Yep. And another very important thing with insects is, let's say we did a study in this meadow back in the 1950s. We were old geezers, and we went through here with nets, and we counted the number of bumblebees and identified them correctly, and it still matches up with today. And we came today on a non-sunny day.
Yeah.
Well, there we've got a decline because I'm not seeing many bumblebees. There's a few hunkered down here. So with insects, you can't just go on one day in the past and one day in the future. You have to do, multiple days or multiple year censuses to sort of see it. And even from season to season. I've been looking at moths at my place near Ithaca I've had my place for almost 6 years now. And every year there's a different time of year where the mothing is just not good. Mm-hmm. There's just, things aren't out, and I can't really explain it, but there's always some point of the year where it's just not good compared to previous years. Mm-hmm. And so if you happen to be there that year, it's like, okay, well, I guess there's no moths in August. Well, no, there's no moths in August this year, but next year will be fantastic.
Yeah.
And there's no moths in August this year at this one spot. Right?
Yes,
Yeah.
Exactly.
Right.
And even like, something as small as moving a light, just as much as like 3 meters, barely any distance, you get totally different stuff.
You mentioned that. I remember, now that you're saying that, I remember that from our previous episode with you, that just a slight movement of the light can change what you get coming into the light.
Different types of light, if you had a different size sheet that the light was at, all these things can factor in.
And that goes back to what you were saying before, how we just need more research, more data from different sites, longer periods of time to have a more robust data set, right?
Absolutely, yeah. And also to write down our methods so they can be repeated,
right.
And I think the final thing I want to mention here, and this is, I was briefly on the Committee on the Status of Endangered Wildlife in Canada, the Arthropod Special Subcommittee, so looking at endangered arthropods in Canada. And my most valuable lesson I got from that is the importance of negative data. So it's one thing to say, yeah, there's no bumblebees down there, but did anybody actually look? And so it's important. And what I like about iNaturalist is I can get data from there where I can say, okay, well, I know this person looks at bumblebees, and they noted all kinds of different wildflowers down there, and they didn't note any bumblebees. So I'm thinking that's some good negative data. They're not there, which is much more powerful than just saying, I don't see any.
Yeah.
This one time.
Yeah.
I brought up that I did insect sweeps a long time ago. It was through the Prairie Research Institute in Illinois. And one of the things they do is, so they have a few hundred sites all throughout Illinois, and every 5 years they cycle through every site. So they have all their data points are, we know what this site was like 5 years ago, we knew 10 years ago, 15 years ago, and so every site, not only do they know the composition of the plant community, given the transects, but along those transects, we've done the insect sweeps. I think it's like 5 meters out from each transect. Or for one of the transects, it was 5 feet out or 5 meters out, and he did the sweeps on either side.
What's a transect, Steve? Just in case people
it's just
don't.
They actually these sites are permanent, so they actually have an anchor in the middle, and then they have degrees that you have to go out in a certain direction, and then there's like another anchor, 50 meters out or whatever, and then you just put a tape measure out, and then every meter along that transect on one side, then the other side, then the other side, then the other side, you identify every plant within I think half meter by half meter square. So that's what the transect is. And we always had to include things like the time we collected, the weather conditions, everything. Because I have to imagine that, because we're only at a site one day every 5 years.
Whoa, it's a drop in the bucket.
It's like I feel like it's got to be so important that they take that data, all the climate data and everything.
All the variables,
Yeah.
But you're also doing multiple sites too, which
yes.
is fantastic. So Illinois is one of the better states for long-term data sets like that.
I'd love to check back in with them and see have we published this data, or what has been done with this data now? Because I only did it for 1 year, so it'd be really interesting to see if they've collected a bunch of it and published it somewhere. So
yeah, so for New York, I've started to get interested in decline in moths, and it's hard to use baseline data here because the people that collected before me, they're not going to keep track of all the common stuff, which when you think about it, that's what's ecologically important biomass-wise. It's the common stuff that's driving things. Yeah. The rare stuff, not so much, but that's what we're interested in. And, a lot of the collectors, if it was a beat-up specimen, I'm not going to collect that. So you've lost all that data there. So for, I don't know if it's good reasons or not, but I decided to start a little pilot project this year at my house where basically I've standardized a sheet and a light and a method where basically I count the common moths. And just to get that baseline data that hopefully someone can use 50 or 100 years from now to compare. Now, of course, there's all kinds of problems with it and whatnot. I'm losing a lot of sleep because it takes a lot of time counting micromoths especially. But it's been very eye-opening for me how my eye was trained to look for the rare stuff. And so I would estimate, okay, I bet there's probably 30 of that one species there, and there'd be 120, Wow. Or I bet there's 10 of that. Oh no, there's only 8. And so, and the other problem is, I'd have to get up at 4 a.m. to beat the catbirds to it. So, I've been very sleep deprived this summer for that reason.
This is what undergrads are for. Why don't you have to get set up with a lab?
And see, that's the problem. I'd love to do that, but it's taken me 5 years to come to a level where I know the moths and I know which ones I can reliably identify. Right. And so it takes experience, For something like butterflies, okay, that's quicker to train. Can't really run a light for butterflies. Some will come, but not many. But there's things like a pollard walk. Have you guys heard of pollard walks before?
No.
It's basically a transect where you count. I'm trying to think now if it's 2 meters on either side of you, any butterfly you see. And it's a standard walk, and you keep track of the weather, sort of like a sweep sample, but you're visually doing it and catching anything you need to. And
cool.
Keeping track of butterflies.
Why is it called a pollard walk?
I'm guessing someone named Pollard invented it. I don't know.
So are you excited for in-the-field researchers to be replaced with robots that can be instantly trained and don't care about being out at 3 a.m. or 4 a.m.
I would love that.
You're okay with it.
I wonder what I'm curious what the audience would think of robots being more ingrained in society, because I kind of don't mind it for certain things,
Well, and it's made being a naturalist a lot easier in many ways. When you use iNaturalist, the AI has gotten so good in the past just few years.
Yeah.
Like the moths at my light, I think I caught 1 error this year, all year.
Wow.
1.
Wow.
It's nailing everything, or it doesn't know. And that's fine. Doesn't know is a good answer. It's not giving me false data. So it's really good. Mm-hmm. And also for a lot of other groups I don't know quite as well, it's coming up with something and I'm like, maybe, and I'll check. Yeah, oh, sure enough, nailed it. So it's the AI is really rapidly becoming useful, or as A1 as some call it.
I talked about this a little bit in our sturgeon episode, but there's been some papers that came out recently with imaging. So typically we have to dissect the fish or do genetic work or whatever, but like just based on pictures, super non-invasive, AI can, with really high accuracy, like above 90% or whatever, determine if it's a male or female, which we can't, a human can't eyeball and do without some kind of invasive method. But, it's a tool, and it's like people who aren't using this tool, I think, are going to fall behind, so...
And, I think back to the days when we were younger and using field guides and sloppily learning things and
right.
Probably making lots of errors because there's no checks for it.
Right.
Pre-internet days or what was on the internet was pretty sparse, and luckily I had a digital camera early on so I could photograph things, and going back and checking things I was confident on. Oh boy, I made a lot of mistakes. I was way overconfident than I should have been.
Dude, I miss the old field guide and camera days. Those were such fun. I mean, also younger, less responsibilities. I could be out in the field all day long. But so I know that there's a lot of things attached to those memories. But it's nice. And someday maybe again. But
Well, Steve, you can always leave the phone at home.
Right? Can I leave my kids at home? I don't know. That's the problem.
Yeah, they're kind of heavy too.
Yeah.
I'd love the day that I can go out with them though. That'll be nice.
Yeah.
So.
Get him his first field guide.
Yeah. But I do remember my first time, because, you grow up on field guides a little bit, and then the first time that I had a job where you had to key things out with Gleason and Cronquist or whatever else, you're like, oh, surely there's only a few hundred species. Oh, no, when you get the actual, estimated species number from Gleason and Cronquist, or, well, that would be the one for our area anyway, so... it's thousands.
Right. So folks, not people...
You're missing on the diversity just by using field guides. So.
People who may not know, when you buy a field guide off a bookstore shelf, they only have so much space.
Right,
they can only list so many species.
It's not like a hardcover flora that belongs in a lab somewhere, So
when Steve mentions Gleason and Cronquist, that's as far as we know, almost a complete flora of a state. So that got me thinking, Jason, I have the Peterson Guide to Insects of the Northeast. So I imagine when you look at that is a tiny sliver of the actual insects that are present, what they can list in that field guide, right?
But they did a really good job of capturing the things you're likely to see and likely to pay attention to. And so it's that great gateway drug, into entomology. And so, as much as there might be inaccuracies or oversimplifications, it gets more people interested. And so I need to mellow out and be less critical because it's bringing more people into natural history.
Yeah, and I feel that that's what iNaturalist is doing as well.
Absolutely.
It's a naturalist in your pocket.
Yep.
We have some ants tending some of these, I can't remember, leaf hoppers, plant hoppers, whatever, these little...
So those would be tree hoppers.
Tree hoppers.
And so you've probably heard of ants tending to aphids for their honeydew.
Yeah.
And they'll take good care of them. They also do that with other insects like tree hoppers. And so those look like some formica ants. And those, if you poke at some of their tree hoppers, they might give you a good sting. You'll feel it. They'll inject some formic acid into you. But yeah, they'll take great care of those aphids there. If you were to cut down that plant, they'll likely pick up those tree hopper nymphs and move them to a new plant. If you attack the tree hoppers, they'll attack you. So those aphids are safe from a lot of predators. So predators have to be really deceptive. There's some things like lacewings where they'll actually take dead aphids or dead plant hoppers or tree hoppers and paste them to their back so they actually don't look suspicious and produce their own secretions so the ants will tend them.
So I got a question here. So way back in the day, speaking of the old camera days, I actually have a picture of a nymph and its tail's up and there's a ball of honeydew on it. And then in the next picture, the ball's gone.
Oh,
and
nice.
It's a really cool series of photos that I have. But right here, there's an ant and it looks like he's tending an adult. Can they get the same treat from an adult? The same honeydew that's on the... Oh, and there's another adult on me.
Well, that is different. That's a spittle bug. So the nymph of that produces little balls of spit on plants.
Are they not in the same? I know they're hemipterans still.
They
right?
are hemipterans, but different
bugs,
families.
But different families, but are they in the same larger group?
Is it so the taxonomy has changed recently. Yes, they're still in that group.
What is that, by the way?
Membricoidea,
I know I said the cicada-ish group, but I always forget.
I think they might be different superfamilies. I could be wrong on that, but definitely different families. And so, yeah, definitely there was an adult.
Yeah, right there.
Yeah, right there. I think that's Encanopin. I think just freshly molted, because normally they're brownish. So that's a very fresh one.
Do they feed from the adults the same way that they can feed from the young?
I don't think so. I think it'll probably skedaddle soon once it's hardened. I bet there's probably some species that break that pattern, but I don't know. But there's actually a lot going on in this one plant here. You see this leaf that's sort of twisted and folded over on itself? Mm-hmm. If you look at the underside, and you were to open up, there's a tiny caterpillar inside there. That's a leaf miner, where it actually cinches the leaf together. It has the lovely name of Cremastobombycia solidiginus.
Oh my God. Solidiginus, though? That's not the first time we've heard that specific epithet.
Yep.
No, what is it, Steve?
Well, Solidago is goldenrod, so it's just related to goldenrod in some way. I can't remember what that ending means, but
yeah, and so you've got so much diversity happening, even with all these ants present, trying to exclude things.
Yeah, just so you guys know, we've been looking at 2 goldenrods this whole conversation.
2 goldenrod stems.
Yeah,
Ants all up and down the stem and the leaves.
And that little white moth is probably Coleophora, which is a case bearer moth, which the cases are hard to find, but a lot of them feed on goldenrod, so its larval case is probably on one of these plants nearby. What's interesting about Coleophora, so a pretty indistinct moth. It was about the size of a mosquito and looks much less impressive. They're very hard to identify. They're much easier to identify by larval case than adults. And the world expert on them, or at least the North American expert on them, basically told me that about 20% of our North American species are described. So 80% are undescribed. It's just daunting.
Amazing.
A lot of work still to be done there.
Yes.
What is that going to take? Genomics as well?
Well, DNA barcoding has been very helpful for that.
Barcoding's huge, yeah.
But it's just daunting,
I feel like it's a different beast taking on insects. In my world, we do a lot of low coverage whole genome sequencing, less barcoding, at least the work that I do anyway.
Yeah, and barcoding is just that course. Well, we should explain the difference between, looking at a genome and a barcode. A DNA barcode is half of one gene, cytochrome C oxidase 1, I believe, and comparing that to other species often delimits the species nicely. So in theory, most species have different CO1 barcodes.
Right. There's initial work that's done to even determine what you should use as the barcode that's specific for the group you're trying to identify, right? So barcode's just a really quick way to get an ID, right?
Yeah, it's kind of like a supermarket barcode where you scan the AC, Gs, and Ts, and it matches that species. Now, of course, it's not that neat. And things that are closely related and hard to identify often don't barcode apart. And they're kind of messy, but sometimes they do. And sometimes you get false positives. Like there's certain moths that I can identify from arm's distance that DNA barcoding can't identify. So it's a wonderful tool to use. But yeah, it's absolutely daunting. And one of the frustrating things dealing with a lot of birders that get bored with birds and move into insects is they expect to be able to identify everything. And boy, do they get really grumpy with me when it's like, it needs dissection. Probably want to be able to ID that. No, sorry, I can't ID that. Well, I found this perfect picture online that matches that. I bet you did.
Right.
Is that identified correctly?
They want to check it off their list, damn it.
And so it takes a while to get through to people new to it that you have to accept that you can't identify everything.
Yeah.
Dude, I might move to birds at some point. So, because I already went from plants to fish. That's like 1/10 of the biodiversity, and I'll go from fish to birds. I'll be like, guys, this is easy.
And then eventually to
bears
echidnas or something.
right. Yeah. I was at a workshop at one point, and these are like the experts in aquatic plants, and it was actually sort of relieving to hear them say. But they're like, there's a couple species of Elodea, but you'll never tell them apart. You probably need genetic data to be able to do it. So just do it to genus, that's fine, unless you're in a very specific area that only one of them is known from. And even this one, there's Potamogetans that look almost identical to it. They have all these different weird leaf shapes. So yeah, you also can't really get this one to species. And just identifying in the field, they're like, these are the limitations. Just be aware, Yeah,
you get to know your local fauna. So I've done my time. I've done lots of dissections. I have a good feel for some of the difficult to ID moths in my backyard. I know what to expect different times of year, but take me somewhere else, I can't do that. I've got to relearn everything from the beginning again.
What do we got here? Hey, you told us not to do that.
I know.
Oh my.
But I also
gosh.
know what this probably is.
What is it?
This is diamondback moth. So this is a non-native moth.
Diamondback?
Yeah. Oh, and it just flew into your face.
Yeah.
Okay, trust me on that. That was a diamondback moth. And they are native to the Old World. They are the biggest pest of cruciferous crops, so broccoli, kale, kohlrabi, cauliflower, cabbage in the world, and one of the biggest insect pests worldwide, and have inspired the use of a lot of insecticides to kill things. And, a lot of negative effects of pesticides that we'll definitely talk about. But that species is now widespread. It's highly migratory here for such a small moth. That's a moth, again, about the size of a mosquito.
Never even heard of it.
In some years, they'll make it to Greenland in good numbers. Whoa, yet they're not frost hardy. So, they did the oops migration. And it's done once. Absolutely huge. And that brings us to an important point with invasive species so they feed on cruciferous crops, but also native crucifers. And there's a lot of endangered crucifers out there, and that's the major herbivore in a lot of them and could potentially threaten some species further. Invasive species can really throw a wrench into things.
Mm-hmm. Wow.
So in terms of the insect apocalypse, have we really talked about the drivers of what's driving insect decline?
So there's a bunch of things, and a lot of the papers point to pesticides as being a very important.
And that's where we'll wrap part 1. Join us in a couple weeks for part 2, where we delve into the details of the insect apocalypse, and we'll also do our regular wrap-up. So thanks for listening, everyone, and we'll see you next time.
Are we actually on the trail?
Are we on a deer trail?
I think we were on an ATV trail.
What's the worst that's going to happen? We find some insects off trail.
Or we not make it back to our vehicle.
That's a good point. Yeah.